I'm Anne Kwitek. I'm another one of the PIs for the Alliance and the PI for the Rat Genome Database, and it's my pleasure to introduce Jeff De Pons today. Jeff is a developer and the technical systems manager here at RGD, and also the Scrum Master for the Alliance. And today he's going to talk to you about JBrowse2, which is the Alliance's dedicated genome browser. So thanks, Jeff. Take it away.
Sounds good. Okay, I'm going to start off with a little bit of background on JBrowse2, what it is. It's the primary genome browser that we've implemented at the Alliance Genome Resources. It can be used to visualize pretty much anything that you can map to the genome. It's an evolution of GBrowse and JBrowse. Those are projects that were started by the Gmod group. I believe GBrowse was about 2002, JBrowse around 2010. And as technology has changed, they've evolved that, and JBrowse2 is a complete rewrite of JBrowse, the original JBrowse, as far as I know, and that's the currently supported genome browser from the group. It's open source software, so you can go download that and browse the source code. I also wanted to point out that there is a lot of really good user documentation available over at the JBrowse site. So if you get stuck or you want to read about how things work, there's help. Also message boards as well. So we'll start off with, how do you find JBrowse at the Alliance? This is our Alliance, genome.org is our URL, our homepage address. And under the Data and Tools menu, you can navigate to JBrowse2. There's also a direct link to it, alliancegenome.org/JBrowse2 listed at the bottom of the screen. When you select that, you're going to be navigated to this JBrowse splash screen. I wanted to put that word on the screen because in JBrowse, there are menus that will, return to splash screen. The word splash screen is used, and this is where you're going to end up if you click that when you're using the tool. I usually start off with an empty session. These are kind of shortcuts to some of the more popular user interface or components of JBrowse, but you can get to the linear genome view or structural variant inspector through an empty session as well. Today we will talk about the linear genome view, and we're not planning to talk about the structural variant inspector. So if I click and start an empty session, I'm going to get this screen here that's going to allow me to in this pull-down menu, it's going to have all the different UI tools that are available in JBrowse2. Today, I'm going to talk about the linear genome view, a dot plot view, and the linear synchrony view. And we're going to start off with the linear genome view. So if I select that and click Launch View, I'm going to get this screen with a pull down of the various assemblies, which correspond to the different model organisms that the Alliance has, a selection for a chromosome, and an open screen. So I'm going to select rat. I'm with the Rat Genome Database, so I'm going to select rat because I'm more familiar with some of the tracks in there, and select open. When I open my view, this is the landing screen that I will end up on. I've got my chromosome is up at the top. There's 250 million base pairs in rat chromosome 1. A box to move change my region to the left and right, which doesn't make as much sense with the whole chromosome, but as you zoom in, that's useful. I can zoom in or zoom out. I can also put a position in this box here or a gene, and it's going to navigate to that position. So I'm going to put a gene in the box, which is LEPR, and from there, it's going to navigate me to the region of LEPR. So you can see up on my chromosome view up at the top where I'm at. I'm around 116 million base pairs. Again, I've got my navigation, but I still don't have any tracks to see. So there's this big button here, Open Track Selector. I wanted to point out this icon. This is very important because there are times in JBrowse where you don't have a button that explicitly says Open Track Selector, and you're looking for a track selector. You can open a track selector in a lot of different contexts. And this little icon here, this hamburger menu with the circles on the left, represents the track selector pretty much anywhere you are. So even if I didn't have this button, I have one right here. That's just important to point out. So if I open the track selector, I get my tracks on the right side. Tracks at are organized in categories and subcategories. So each of these, I can open them up and I may see subcategories or individual tracks. There's also this 3-dot menu. This is a menu here. It's not a more type thing. It's an actual menu where you can do various things with those as well. The top area here are the Alliance value add tracks. So these are tracks that the Alliance has added. The tracks below that are mod specific tracks. Oh, I went one too far. So the Alliance tracks at the top, you've got reference sequence, all genes, variants. We're going to talk about each one of these individually in the following slides. High throughput variants and multiple variant alleles, along with Alliance orthology. And then again, these tracks at the bottom have been developed at the individual mods and imported into the Alliance. So it allows you to, it's convenient to have them all in the Alliance JBrowse. You don't have to go to 7 different locations to get them. In addition, you can open them in JBrowse alongside the Alliance added tracks. So it's, but because they're supplied by the mods, they may vary, anything below Alliance orthology. This is just a screenshot of all the top level categories of the tracks available for the different organisms. So there are hundreds, if not thousands, of tracks available, dependent upon the organism that you're looking at and what you're trying to do, including expression and variation and all sorts of stuff. So we're going to start off with the focusing on the Alliance tracks. We're going to start off with the reference sequence track. So if I select that, I wanted to put this slide on here as well. So it's another common UI component of JBrowse is if you try to open a track that has too many widgets for the screen. This is all in JavaScript running in the client browser. So there are times when it'll ask you to either zoom in or to force load. And I just wanted to point that out. So I'm zoomed out too far to see the reference sequence here. But as I drill in, I will start to see colors on my track. So this is stop codons and start codons, along with a different color for each of the nucleotides. And if I zoom in even further, I'll start to see the amino acid translations for the different reading frames, along with the nucleotide sequence. For context, I opened a variant track and put it alongside the reference sequence. So JBrowse2, and as you'll see moving forward, we can open as many tracks as we want and view them compared to each other. Next, I'm going to move to the all genes track that you'll see for each of the organisms. But I first wanted to add in a couple pro tips about using JBrowse2, things that I've learned over the years of working with this. By default, the track labels overlap the track. So there is an option in this little hamburger menu up here to go to track labels and make them offset to move. You can see LEPROT is covered up by the label here. And then on the track labels, there's a 3-dot menu. And if I select that, I have options to turn the labels off or turn the descriptions off or compact it or collapse it or set the max height. Sometimes you want to change the height of the track that you're seeing. A whole bunch of options to make the view easier to digest. And this is more important and less important with something like this, but more important with something like a variance where you can have a lot of variance if your region is not real small. So we're going to go back to opening up the all genes track. And this is the display we're going to see. So just to give you the landscape, it's very similar to what we described before, but I can drag and drop in the bar here to change the size of my region. I've got each of the genes listed out with all the isoforms, the direction that the gene is going, and that's also denoted by the color. If I select one of the genes, I'm going to get a sidebar menu with all the metadata about that gene. So in this case, I've got the position, the description, the name, the length, etc., external identifiers that are used, and then a link to the Alliance alliancegenome.org site. So if you want to view the gene report for this gene, you can just click here and get there. Then each of the subfeatures, so each of the isoforms and transcripts and exons and whatnot are all listed out in the sidebar alongside this show feature sequence button, which is actually kind of cool. If I select that, I'll get an option to, I can show the CDS, I can the translated protein, I can get genomic with the introns included. If I want 500 base pairs up and downstream, whatever I want, I can choose that, and it will display that sequence for me to be able to pull that out, and that's for any of the genomic elements that you see in the Alliance JBrowse tracks. Moving on to the variants track, this is I've always felt like this could have a little better name. It just says variants, but at least in the context, I think there are plans to add more variants. And actually, if you want to learn about that, variants were the topic of the Alliance office hours in February. So you can go to our YouTube site and view that presentation to learn how the Alliance deals with variants and what variants are included. But this is the phenotypic, these are variants that are associated with phenotypic alleles in the Alliance genome.org. And I wanted to point out as well that each of these variants also have a corresponding allele variant page on the Alliance site. So you can always go get more information there as well. And then I've got a link at the bottom to our YouTube page. So if I open up the variants track, I've got my gene track still open. I can see LAPR has a deletion and SNV with a phenotypic allele. If I select that variant, I'm again going to get my metadata on the right side. In this case, we've got the position, we've got the description of the change, the name. Wanted to point this out. All of the variants, whether they're phenotypic alleles, whether they're high throughput, any variant that you see here is run through the ensemble VEP. So this is also a value add that the Alliance is adding, and you'll get transcript level consequences from the VEP. So I've got it listed multiple times because there are multiple transcripts here. And then down below, you can see since these are phenotypic alleles, you can get the allele symbol. You can get the gene ID of the gene that the allele is part of, et cetera. And then the feature sequence genomic stuff is on the right as well. So I can pull that variant out and I can get 500 base pairs up and downstream. I did notice that this did not put the variant in there. You'd have to take that C and stick it in there, but it's in green there. It shows you where the variant would be in that sequence. I'm briefly going to touch, this is very similar to the phenotypic alleles, but there's a separate track for multivariant alleles. There's only a couple mods. I believe, I don't want to say, I know mouse is one of them because this is an MGI, but there's either worm or fly. One of the other two mods that have these multivariant allele tracks, and these are just alleles that have multiple variants associated. If I select that, I can view the individual variants as well in the sidebar. Next, we're going to jump to the high throughput variants. Now, these variant tracks are associated with whole genome sequencing experiments or public variant repositories such as ClinVar for human or EVA for some of the model organisms. All these phenotypic alleles and high throughput variants are submitted from the member mods, so they may vary a little bit in the sources. And again, I'm going to bring up that February office hours on variants. So if you want more information on where those are coming from and why they were selected to be pulled in, you can view that presentation. So if I open that track, you can see I've got a whole gene here, so it's a really big region. I could scroll down for quite a ways. And this is where this 3-dot menu comes into play as well to kind of make this a little more digestible. I can turn those labels off and I can tighten it up and I can shift it up and I can do all sorts of stuff to make this look a little easier to understand. If I zoom in, that's another way to deal with that is to zoom in. So I've just zoomed into LAPR a little bit, and you can see that this variant is in an exon. These two are not. You can move around each of these tracks too by dragging. If I select the variant, I'm going to get my side menu again, which in this case, you've got the RS ID, if there is one that exists. And then we've got this pipe delimited kind of blob of information, which is, I believe, output of some of the output of the VEP. But this information is actually used over to build this table. And this is actually really neat. It's a 39-column table. I can select the columns here and limit it to just the columns I want. I can filter on this. I can change the density of the table. I can export it as a comma delimited file. And it's all the information about that variant with respect to each of the transcripts. So this is a listing of the columns of that table because I couldn't get it on one screen, but you've got the protein translation, the protein position, the polyphen prediction, CYP prediction, transcript information. There's all sorts of stuff in here. I did not find like a help menu on exactly what these mean. Most of them are pretty self-explanatory, but if you have any questions about that, you could always email us as well. So I thought it was a kind of a pretty cool feature, and that's on all of the variants that the high throughput variants that you see in the Alliance JBrowse. Moving on from that, we're going to go to the last section here, which is Alliance orthology. So this is all added by the Alliance as well. So if I open that, I'm going to see some subcategories, which are stringent, non-moderate, reciprocal best hits, within each of those subcategories, I've got tracks which map the organism that you're on to all the other organisms. In this case, I was on rat, so I've got rat to xenopus, rat to human, rat to zebrafish, etc. And what do those terms mean? Stringent is where an ortholog has been called by 3 or more of the orthology methods, and if it's a best count or a best reverse count. There's the secondary criteria, as if it was called by ZFin, HGNC, or ZenBase, which are manual. So they're manually assigned, which is why they're included. Moderate would be if it's been called by 3 or more algorithms, and period. It doesn't matter if it's the best or best reverse, best hit or the best reverse status. None is going to include any ortholog that's been called by any of the algorithms. And then the reciprocal best hits is if it's the best from mouse to rat and rat to mouse, or from one species to another. Another point here, the next office hours, next month in June, is going to be on orthology and pairology at the Alliance. So I'm guessing this will be one of the topics they talk about there. So I would recommend, if you're interested in this, to go to that next office hours in 1 month. We use only orthology algorithms that have been approved by the Quest for Orthologs, and I just listed out the algorithms that are used. You can see the 3 manual ones down on the bottom right, and these are the algorithms to get that, if 3 or more stuff working. So I'm going to select rat to xenopus tropicalis and rat to mouse from the stringent bucket in the Alliance Orthology, and I'm back on my linear view. And I've got my gene track across the top. I now have 2 additional tracks down below. These bars are showing you. These bars are showing you where there is orthology between this species and this species. So if I look over here, it might be interesting that this gene looks like has some orthology in mouse, but does not have it in xenopus. I can then hover over these, and it's going to show me what the corresponding gene is, or the orthologous gene, or I can click on it, and I'm going to get my sidebar again with my metadata and my information about it. So you can see I'm hovering over that. I see it's LAPR. LAPR in rat. It's also, the symbol is also LAPR in xenopus. The length is 188,000 base pairs versus 39,000. More information on the mating. I can get the feature sequence, as we showed before. And then down at the bottom, there's this launch new linear synteny view on this feature. So this goes back to the linear synteny view tool. I'm going to do that next. I'm going to do it a little different way, because there are multiple ways to get there. I'm going to right click on this bar, and I can also say launch synteny view for this position. If I do that, my linear synteny view comes up. This is a menu if you're hovering over the synteny line here. It's not very interesting that it's just one gene to one gene. It's running all in the same direction. But I can hover over that, and I can get information about the 2 orthologs. I can also click on it, get my side menu, which is going to be very similar information. So what I did then is I zoomed out and scrolled over a little bit, just to try to look for something a little more interesting. And it looks like this region's all inverted. You can see by the crossing here of the synteny lines. But you can hover over any of these bars. I can go to the top, and I can drag to reduce my region down. I've got an input box for the position up at the top, which I can add genes in there. So if I wanted to set a gene here or gene here, I can do that. I can drag and drop these tracks to move them left to right, and all my lines are going to stay connected and kind of flow. I can also, here's an example of the track selector. If I wanted to add tracks, that's where this gets really powerful, is adding additional tracks for variance or expression or whatever it is that you're looking at. Here's my track selector button up at the top, which is that menu with the 3 circles on the left of it. So if I select that, I could add tracks to either side of this. So the linear synteny view, which we were just looking at, was embedded. So we created an embedded linear synteny view. There's also the linear synteny view tool, which is very similar. I'm going to go back. I went back to the splash screen, started an empty project, and I can add as many species as I want here. I could have 4 or 5 and view the orthology between all of them. One little caveat, and this is just kind of interesting, and I thought I'd mention, is if you add for each species that you add here, you got to click this little arrow. So if I add 3 of them and I don't click this, I'm only going to see the top two. It's just a very strange behavior that I thought I'd mention because I struggled with it a little bit at one point. So you click the arrows, and then I'm going to launch that. And here's what I'm going to be shown. It's the entire genome, all the chromosomes listed for rat, mouse, xenopus, along with all the orthology lines. I can open a track selector for each one of them. I can go up to the top again and add genes in each one, or change the region if I wanted to change. There's a box for each of the species. In this case, I'm going to add LEPR into for mouse. So I set LEPR in for mouse, so it changed my region just for mouse. I've still got all my chromosomes for rat and for xenopus, and you can kind of see the orthologies all going to chromosome 5 in rat and xenopus chromosome 4. So I can scroll down, and you can drag and drop in here if I want to just reduce my region down, or again, go edit my positions from that point. Here I opened a track selector and just opened the genes for each one of them and reduced my region down. And you can see, LEPR's here. Here's my line of orthology, and then here's LEPR, and then it's back over here. Here's LEPR. And again, these you can drag them back and forth, move them back and forth. I can open individual tracks for each one of these things. And with, try to comparing, rat to xenopus theoretically through mouse, theoretically, if I wanted to. So here's an example of I just opened a couple tracks alongside it. Got human genes. I opened the human high throughput variants, and this is orthologous to this region here. You can see that there's actually an inversion there is not between human and xenopus, but there is between xenopus and rat. There's an inversion. And then at the bottom, I've got a strain-specific variant, damaging variant track open for DA-OLA HSD, which is a model for a variety of things, but one of them being rheumatoid arthritis. Next thing I wanted to go through, I'm not going to spend a lot of time on, is the dot plot view because it works in here. If I go back to the splash screen and then select view to launch, select my dot plot, I can select it's a whole genome comparison tool. So I'm two assemblies. So select Xenopus laevis and Xenopus tropicalis and launch my view, and it's going to give me this plot of the two assemblies. There was a duplication event in Xenopus, which is why I chose these two. So chromosome 5 maps to chromosome 5S in addition to 5L. You can also see over on chromosome 9, there's an inversion here. So it's going the other direction. But you could do this for rat versus mouse, or rat versus human, or a fly versus worm, or whatever assemblies you'd like. I wanted to add in a little nice to have feature. There is the ability to add your own tracks in JBrowse. So you can use the built-in tracks, but I can go up to this add a track selector. There's connections here to USC Track Hub if you'd like to pull something in it from a public repo, or I can enter a URL in here if I've got an HTTP URL or an FTP URL, or click File and upload a track into my my private track so I can compare with the tracks that are on the screen. The supported data formats, a lot of bioinformatic formats, Big Bed, Big Wig, CRAM, BAM, VCF, GFF3 are supported for those tracks. Last thing is just to, this may seem obvious, but I just think it's kind of neat that I can go up and add I can add a linear genome view and then a dot plot view, or multiple linear genome views. So in this case, I just added, it's a gene track for rat and a protein track for worm, and then a zebrafish knockdown reagent track, which are unrelated, but just the point that you can add anything you want all in your view, which makes it kind of nice for building screenshots or whatnot. It doesn't have to be necessarily species or even track type specific in your view. With that, I'm going to move on to acknowledgements. Scott Kane, who's on the call today, did a lot of the work on this. He's kind of rolled off it a little bit, but he's did the majority of getting this set up from Penn State. And then Paul Hale from the Jackson Laboratory is taking over some of the maintainer role for JBrowse. Also wanted to thank the data core masters at the Alliance. There are groups of programmers and curators that work really hard to get all the data from the mods into the Alliance so it can be harmonized. And with that, I'm going to take questions. I do want to mention real quick before we do questions that there is next month on June 12th, we'll do Orthology and Paralogy at the Alliance presented by Chris Tabone. You can go to the Alliance website. So under news, there's an events calendar. If you forget where it is, you can register for that. And we hope to see you all there. But we will go back to the questions slide.
Thank you, Jeff. If there's any questions, you can unmute yourself or put your question in the chat. Jeff, one question I had, and this might be more for Scott as well, is there are different ways of defining conserved synteny, right? There's different algorithms that people have developed to identify and define the boundaries of these conserved synthetic regions. Is there an ability to change which method of defining the synteny boundaries is being used in the display?
So yeah, I guess I'll go ahead and answer. So basically, what the Alliance JBrowse2 has for synteny / orthology is with every release, the build process builds this gigantic orthology file where brings all these pairwise information of this mouse gene is orthologous to this rat gene, and here's why we think so. And so basically, along with that build, the JBrowse part of the build looks at that specific file that is generated for the build and pulls out all that information and puts it into the into JBrowse so that then it can be used for these displays. It would certainly be possible if somebody wanted to generate their own set of data. So basically, what JBrowse ends up with after it parses through that orthology file that the Alliance builds is basically just for each level of stringency and for each pair of assemblies is basically just a list of this gene versus that gene in the two different assemblies. So you could very easily create something different. For instance, you could run some sort of other tool that will generate, say, alignments and would give you synteny. So for instance, if you wanted to, you could run Minimap2 and compare rat and mouse and put the Minimap2 results into JBrowse. And you can do that. Jeff mentioned that you could add your own data. You can add your own synteny data as well. Okay. So you could run an analysis and generate your own synteny track between whatever assemblies you want to do. So yeah.
Great, thanks, Scott.
Yep.
We do have a comment in the chat from Zenbase. Thanks for covering Xenopus so well. That's good to hear. Always good to get good feedback. Any questions or comments from anybody else?
Hi, this is Sean. I actually tried using JBrowse at the Alliance a couple of weeks ago and just bounced off it, which was embarrassing because I've been using GBrowse and JBrowse for years. And I want to say this talk really led me through where I was going wrong and trying to do anything at all.
I have heard that it's extremely powerful, but there are a lot of menus all over the place. And if you get comfortable with where things are, it can be daunting in the beginning, this tool.
Yeah, it's like I've been using JBrowse at Flybase for literally decades. So thank you for helping.
Yeah, I sympathize because JBrowse2 is kind of a reimagining of what a genome browser can be, right? I mean, there's up until JBrowse2, a genome browser was a line that had colored boxes on it, basically. And JBrowse2 is so much more than that. So but that comes with an increase of complexity for the user, and so yeah, I agree, Jeff, you did a great job covering just a few aspects of what JBrowse can do, because like he said, there are there are multiple things under that initial menu that you can do that we didn't even talk about. Yeah.
Yeah, and I want to mention, and maybe you can go off of this, Scott, there's all sorts of additional widgets apparently that you can add in. Is that true?
Well, I don't know. I guess it depends on what you mean by widgets. I mean, so like there's a really neat breakpoint inspector. So if you think you have say you've sequenced a strain of worm, and you think that you've got some chromosomal rearrangements, if you've got BAM files, you can then use that to actually inspect what the BAM files do, like around a breakpoint, and so it does a really nice job of drawing lines between, oh, this read on chromosome 1 actually, jumps the shark and then ends up finishing on chromosome 2. So, so it gives you a really nice visual display of breakpoints. So that's pretty neat. But yeah, I guess I guess I don't really know what you mean by widgets. If you give me an
I
example.
meant plugins. So if you go up
It's
to
the
the tools
plugins.
menu,
Oh.
there's this whole thing of plugins.
Oh
for-
yeah,
on the
absolutely.
floor.
Yeah, and most of those are to give you kind of new or different track types. Being able to display different types of data. I can't think of any off the top of my head because I haven't
There's
looked
one for
recently.
multi-alignment files, which I think might be helpful with some of the Synteny stuff as
Yeah.
well, right?
Yeah, so yeah, that'll let you. But yeah, what's neat about that one is, yeah, it'll let you display. So say you have a multi-alignment, yeah, you can display it all as one track, so you can see basically all of these alignments kind of lined up against a given chromosome, so that you can see.
Yep.
I'm trying to think of a good example. I can't think of any, but yeah, there are a lot of neat things. Yeah.
There's a steep learning curve, but once you kind of get the hang of it, it can
Yeah.
do a whole lot.
All right. Well, thank you, everybody. Anne, any final words from you?
I don't think so. Thank you all for coming, and thank you, Jeff. This has been really helpful.
All right,
Thank
great.
you.
Thanks, everybody. We look forward to seeing you at our next office hours in June.